For the PCR mix, you will need several components. These are contained in the master mix provided by your TA. This one contains nucleotides, tag polymers, magnesium ions, and buffer. Start by labeling the side of your PCR microcentrifuge tube with your seat number. Add the master mix to your tube according to the protocol. Now add your extracted DNA to the PCR tube. This volume will differ depending on the concentration of DNA in the extraction, so make sure your calculations are correct. Mix by slowly pipetting up and down, being careful not to make bubbles. Now you are ready to start the PCR reaction. Using a thermocycler makes the PCR reaction much easier because it will automatically change the temperatures according to whatever protocol you design. Ask your TA for assistance if you do not know how to set the proper protocol. After the PCR reaction is finished, you need to determine if you obtained an amplified DNA fragment, which will be visible on the agarose gel. This first step in preparing the agarose gel requires these components: buffer, not water, agarose, and DNA dye. Tare your scale for proper measurement. Then using the spatula, weigh out the designated amount of agarose. Place this all in the flask. Next, add the TAE buffer to the proper level. Microwave the agarose mixture in short increments, swirling the contents in between to check until it becomes clear and the agarose is dissolved. When the solution boils and is completely clear, it is finished. After the agarose has cooled slightly, you will need to add the gel star stain. The stain is what makes it possible to visualize the DNA under UV light after we run the gel. While the gel is cooling, you need to tape the ends of the casting tray. Taping the ends prevents the agarose from leaking out while the gel is cooling. Be sure to push hard and use 2 layers so the seal is tight. Then place the comb, which makes the wells, where you will put your sample, into the gel about 1 centimeter from the end of the gel. The agarose should not be poured directly after being melted. Let it cool until it is lukewarm. You will need to put enough agarose so the bottom of the wells are completely submerged, about 1 centimeter deep. It will take about 10 minutes to set at room temperature. When the gel is ready, it will no longer be clear. Carefully remove the comb to expose the wells by pulling the comb straight up and out of the gel. Then remove the tape and both ends of the gel. When you place the casting tray into the electrophoresis chamber, orient the wells at the black negativity, charged cathode end of the chamber. Now pour 1 times TAE buffer until the entire gel surface is submerged. To load our samples into the wells, we add loading dye. This dye contains glycerol, which helps with density of the sample and allows tracking of the migrating DNA. To mix this dye, use a small portion of parafilm. First pipette the designated PCR sample into the parafilm. Then add the loading dye and mix by pipetting up and down, careful to not make bubbles. A ladder is also needed, so add DNA ladder to the parafilm and another portion of loading dye. Do not mix ladder with your sample. Pipette the designated amount of PCR sample and ladder by placing your pipette tip slightly down into the well. Be careful not to rip the gel or stab the bottom of the well. Slowly dispense, then remove the tip from the well. Put the lid on the electrophoresis chamber, ensuring that the wells are on the negative cathode end and the poles are placed correctly. Connect leads to the power supply and set for approximately 110 volts. If done correctly, bubbles should rise from the leads. Stop the gel when the blue tracking dye is approximately 1.5 centimeters from the end of the gel. Turn off the power supply and disconnect the leads, then place the gel on an imaging tray. Slide the imaging tray into the imager. Then select the gel star settings and take a picture. After, discard the gel into the trash. Rinse the tray with water and dry it with Kim wipes. If the PCR reaction was successful, you need to remove excess nucleos